And in this endeavor, the American Center played the key role, and we thank American Center for their support for bringing her to India and to Calcutta. And to introduce her, I request Mr. Jonathan Ward, Principal Commercial Officer of the American Consulate, to please come on stage and introduce Dr. Bhattacharya.
Namaskar. How is everybody? Are awake? If you're not awake now, I'm going to make sure you are. My name is John. That's all you're going to know about me, because I am not the important person here today from the United States. We have the great privilege to have a very special guest. Have any of you guys seen the movie Hidden Figures? The story about some great ladies that had such an important role at NASA. Our guest today, is a similar, very accomplished lady. In fact, I heard a story. I actually read a story, Dr. Bhattacharya, and it's in the public, so I think I'm allowed to repeat it. When Sharmila was just a little girl, she asked her father, who was a pilot with Indian Airlines, can I be a pilot? And her father, being the great, inspiration that I can tell that he was to your life, he said, well, of course you can be a pilot. You can be president of the country if you want to. So Dr. Bhattacharya is a true inspiration for all students, but I think especially for our girl students here today. And it's great to see so many of you here. In the United States, and I think you have the same saying, if something isn't hard, we say, well, that's not space science. Well, here we have a true living and breathing space scientist here with her family, and I know that her mother and other relatives are so very proud of what Dr. Bhattacharya has accomplished in the United States. Now, I won't go into great details about some of the scientific investigations and breakthroughs, because even though I have some training as a scientist, I really don't understand some of the work that you've done. That's how advanced it is. And please let this be an example for what all of you guys can accomplish if you set your minds to it. Because Dr. Bhattacharya grew up here in Calcutta, went to college for a few years at Presidency before going to the United States to further her education at some of the top universities in the United States of America. So as a commercial officer with the consulate, if I can put in a plug, we would love to have you come to the United States and study as well. Now, what I would like to also thank Dr. Islam and the entire staff here at the Birla Museum. I'd like to recognize Shamar Bagchi, who I just had the privilege to meet, who was the director here for about 20 years, 29 total years with the Science Museums. The National Council of Science Museums, which is based here in Calcutta, is a great friend of the U.S. Consulate and the U.S. government. If you've gone to a science city and seen science on a sphere, that technology was developed at my agency, the U.S. Department of Commerce, the National Oceanographic and Atmospheric Administration. So our governments work very, very closely on science and on science education. Of course, the National Aeronautics and Space Administration, or NASA, where Dr. Bhattacharya works, has worked closely with ISRO, and we hope can work more closely in the future. Of course, the accomplishments of ISRO and the scientific community in India are just growing every single day. Dr. Bhattacharya came to India on this visit for an official visit at a conference, the International Conference on Radiation Research, put together by the University of Hyderabad. So we hope Dr. Bhattacharya, that we'll see you again in India many times, and welcome home to Calcutta. So without further ado, would you everybody please join me in welcoming Dr. Sharmila Bhattacharya.
প্রজেক্টরটা চালিয়ে দিতে বলুন নোটিস আছে প্রজেক্টরটা অন করে দাও
ঠিক সেট করব হয়ে গেছে প্রজেক্টরটা নাড়িয়ে দিয়েছে দেখুন দেখেন
No, I don't.
Well, firstly, thank you so much for being here. It really is, and thank you to BITM, thank you to the American Center for organizing this talk. But I was particularly excited to come, and when I was invited to come and speak here in Kolkata, I was really excited because I owe this city a huge debt of gratitude. This is where I grew up, as John mentioned. This is where I did all of my early education. And many of you students in the audience, I remember being in those seats, well, I shouldn't say not so long ago, long time ago, but I really think that this is where I developed the excitement for the science and for the work that I then later went on to do. So thank you again for being here. Thanks for all of the inspiration. So I'm going to talk to you a little bit today about the endeavor, the effort where we as a humankind, we are prone to want to explore new places, new things, discovery. And so one of the efforts that NASA has, and in fact, in time, I'm sure many of the space agencies across the world, is to go into and explore deep space, go beyond where we've been before. And so in order to do that, as a scientist, you want to understand many of the parameters. How will the environment affect you? What do you do in order to make it a safe environment for you to be there? So in order to do that, we scientists, so as a biologist, one of the areas that I'm interested in is how is a biological system affected by this environment? And so today, I'm going to divide my talk into two parts. The first part, I'm going to talk to you about a deep space mission that we're building now, that we're planning and we're building it, and I'll tell you all about it. It will fly on a satellite in 2019 to 2020 timeframe, and it will go around the sun. So that will be a very deep environment. We have never sent biology to those altitudes, and so that's the first thing I'll tell you about. The second part of the experiment, or the second part of my talk, I'll talk to you a little bit about a couple of experiments. One that we've done already and have the data for, and one that we just finished doing, where we use fruit flies. So these are the little tiny fruit flies that buzz around your bananas, your rotting bananas, but they're actually an extremely useful tool for science and for biology. And so I'll tell you a little bit about how you can use that to do valuable science in space. So let me orient you a little bit. When I say deep space, what do I mean? So if this were the, so this is the surface of the Earth. This is the International Space Station. So this is that multinational collaboration, groups from the US, from Canada, from Europe, from Japan. Many people have worked together to build and make this space station a platform in the sky where you can do science. And so many of the experiments that I'll talk to you in the second half of my talk with the slides are actually being done on the International Space Station. The other, so what I'm going to talk to you at the beginning, the satellite mission, that's actually going to go much deeper. It's going to go the other way, but it's going to go much deeper than this area. Why is that something that we need to think about? Because there's something called a Van Allen belt. It's a magnetic sphere. Think of it as a shell that protects us on Earth from the damaging radiation that comes from deep space. And so if you were actually going to go to the moon, which is at about, let's say, 85,000 kilometers from the surface of the Earth, you would have to cross that protective shell and you would be exposed to radiation. And then as you go deeper, if you went to Mars, it would take you 3 years to go to Mars and come back. And so in that time, you would definitely be exposed to a lot of this radiation. So NASA, along with, I'm sure, others in the future, so we're developing a capability called the Space Launch System. It's a heavy lift rocket capability that will allow us to take experiments and people into deep space. And so as that is being developed, side by side as scientists, we are working to understand what these environments will do to biology. So when I talk about radiation, what exactly am I talking about? What do I mean and why is it so harmful? So there's two kinds of radiation that you worry about when you go into deep space. One is called the galactic cosmic radiation. This is, these are heavily ionized charged particles that are from within our galaxy, but from outside our solar system. So they're coming from pretty far away, but it's like rain. It's a continuous onslaught of these particles, like iron, carbon, oxygen, that are highly charged. And when they penetrate into the cells of your body, for example, they could cause a lot of damage. They could damage your DNA, they could mutate it, and so on. And they're also very hard to shield against. Some of you in college will start doing experiments with radioisotopes like phosphorus-32, but you can shield against those. You can work behind the shield. With galactic cosmic radiation, it's very, very difficult to shield because these are they're like tiny, tiny microscopic cannonballs. When they come in and hit the body, they will cause damage, and it's very hard to block it. The second kind of particles are solar particle events. These are protons. These are not as massive, and they are much easier to shield against. But the caveat for them is that they're very unpredictable. So you don't know. So when, let's say, an astronaut is out there fixing something on the International Space Station or on another mission into deeper space, when they step out into space, if an SPE hits, they have no warning. And the radiation environment can be almost 1,000 times greater than what it normally is. And so, so there are these kinds of particles that we are studying, and we want to make sure that we have a plan and a strategy to mitigate against. So what are the limits of our knowledge so far? Well, the studies that we've done so far, and I'm talking even historically speaking, we had 12.5-day trips to the moon. So in other words, there were Apollo missions where we know astronauts went to the moon or went around the moon, and those were beyond this protective magnetic sphere. So they were exposed to radiation, but the moon is not so far from Earth. So those missions were only a week to 2 weeks long, right? So that automatically limits how much radiation that you're going to encounter. The second kind of missions that we've been doing actually quite frequently, internationally speaking, we've been going to the International Space Station, and sometimes astronauts stay as long as 1.5 years. So that's wonderful, but because that gives you the long duration, but the ISS is at 350 kilometers from the Earth's surface. So it's inside that protective shell. So you don't have as much radiation as you do when you go beyond this. And so clearly there's stuff that we need to find out because we don't have all of that information on hand at this point in time. So before I go on and explain some of the other stuff that I'm going to talk about, I wanted to, for those of you in the audience, maybe you haven't yet got to this part in biology in your coursework, or for others who may have forgotten, I wanted to just give you a little bit of an orientation to the biology of the system that's important to us. So DNA, what is DNA? DNA is this genetic, it's really a chemical molecule that's inside every one of your 1 billion cells in your body. And it's the genetic material that actually carries all the information from your parents to you, and that someday you will transmit to your children and grandchildren and so on. So it's our inherited material that's present in all the cells of our body. So you can imagine it's an extremely important molecule, the DNA. The DNA then is organized into these discrete structures. So we have 23 pairs or 46 pairs in each of our cells, and these are called chromosomes. So the chromosomes are bundles of DNA that are present inside each of your cells. What is a gene? So the chromosome, if you think about it as a sentence with words in it, then the gene is like one word. So gene is a unit within the DNA that gives you information that you can understand. So altogether, all of the genes, when they're expressed in your cell, they're the ones that are making you think right now. They're the ones that are allowing you to see the screen. They're allowing you to listen and to hear, to understand what is being said around you. And so these genes, what happens is the genes are what is called expressed. So the genes code for proteins. So actually, Hargobind Khurana from India, who won the Nobel Prize, actually worked this out. And so the genes make proteins, and these proteins are the workhorses in your cells. So your skin is a protein. The enzymes that you secrete to digest your breakfast, those are proteins. So the proteins are the ones that do all the work in your body. The DNA carries all the information. The DNA then makes protein that does all the functions in your body. Now, what is a mutation? So a mutation is when you have either radiation or other toxic insults in the environment can affect your body, and they can cause a change in your DNA information. And when it does that, these genes that are coding for those proteins that are going to work in your body, those genes could be altered. And so then the protein that's made is different than what it should be. And so then you can get a messed up function in your cell, and that's not so good. And that's what a mutation is. And so ionizing radiation, which is a kind of radiation that I'm talking about today, can damage DNA and can therefore ultimately result in cancer in cells. So some historical data. In spaceflight, so this is an experiment that was done back in the 80s, they used this insect called the stick insect. Some of you may have even seen it in your garden. There's this long stick-like, it looks like a cane stick. When they flew that into space, what they found is that in the insects that had been hit by radiation and that were in this environment with no gravity, remember in space flight, there's two things that are changing. You have no radiation, you have no gravity and you have radiation, right? So in the insects that were hit by radiation in this lack of gravity environment, they had some abnormalities or anomalies in their development. So some of these stick insects, which are so straight, they have these curved abdomens, or their antenna, which is straight, which is used to monitor their environment, had these weird curved appearance. So again, from some early studies, it was already becoming clear that the combination of radiation and no gravity could have an effect on these organisms, these multicellular organisms. Another organism, the fruit fly, and I'll tell you more about it in the second half of my talk. An experiment was done in the 90s where it was seen that when these fruit flies were sent into space and brought back down, the offspring, so the children and so on of these fruit flies, they inherited some mutations. Remember I mentioned to you the DNA, and if you change that DNA, you can mutate the DNA, and then you can affect the function of your cell. So some of these fruit flies that came back from space, in their reproductory cells, they had some of these mutations, which they then transmitted to the next generation. And so that's something also, obviously, that, as biologists, we are interested in looking at. What about plants? So with these simple mustard plants that you'll see, everywhere, they almost grow like weeds. An experiment was done for 6 years, which is a really long time. Still in low Earth orbit, still within the protective environment of this magnetic sphere that's around us, but still 6-year experiment. That's probably one of the longest biology experiments that have been done so far in space. What they found is that these mustard seeds, when they brought it back to the ground and they germinated them, there was a reduced frequency of germination. So many fewer of the seeds actually germinated into a plant. And also what they found is those that did germinate into a plant had delayed germination. So they were latent. It took them longer to get out of the seed stage. And there was also increased mutation frequency. So all of this points towards the fact that there can be changes in a biological system. One of the things I wanted to point out here also is that every time someone does the experiment, it's coming from different places. People are using different hardware. One's coming from the university, one's coming from somewhere else. They want to use their favorite plant or their favorite insect. So what happens is we have some data out there in the scientific literature, but they're so disparate, they're so different, that it's sometimes very hard to compare from one experiment to another, and therefore compare the effects of radiation. So our idea, our concept is that we want to come up with a standardized experiment, a standardized what we call a payload. So an experiment that you fly in space is called a payload. So come up with a standardized payload that's small, self-contained, automated, and reproducible, that you can then fly to the International Space Station. You can fly the same thing to Mars, you can fly the same thing to the Moon, and then you can compare the biological response that you will get from each of these environments, and then start to conclude and predict and estimate what effect biology will have in that particular orbit. What about astronauts, right? So I showed you insect changes, plant changes. What about humans? What do we know about that? Well, so when we take astronauts, either those who have been on the International Space Station or cosmonauts who have been on the Mir Space Station, and you take the data of all of those who've been there for 6 months in those low Earth orbits, and you take their blood sample before they flew and after they come back from flight. And then you do an analysis. You take the cells from the blood, you look at the DNA, and you look to those chromosomes I told you about. You look at the chromosomes, and you see what changes have happened in those blood cells after flight as a result of flight. And basically what you find is that there's almost a 1.5 to 1.8 times greater number of abnormalities in the chromosomes in the blood of those astronauts that are coming back from a 6-month mission in space. And also the other interesting thing is that more than 80% of those changes are attributed to this galactic cosmic radiation I told you about, the heavily ionizing one that's very hard to shield against, the very damaging kind of radiation. That's most likely what is causing this change. So we just recently published a paper that's out of the open literature where we looked at, if you looked at the damage I told you about in astronauts on the International Space Station, which again, remember, is a low Earth orbit, it's a little closer to Earth's surface, and you compare it with what you would estimate would happen when you went on a trip, let's say, to Mars and back, or some deep space what you find is in the same time frame, let's say a little under 2 years, on the ISS you have a certain amount of damage, but you get 4 times as much damage at the same time duration when you went beyond Earth's magnetic sphere. So again, this is something that different cells, different organisms all seem to have or show a response to this radiation. So then, this ionizing radiation, I've already told you, may present a challenge to humans. We have to prepare for long-term human exploration. We as humankind are most likely going to get there. And so before we do, there are some studies we need to do. There's a critical knowledge gap. We don't know that much about what this environment is going to do to your cells. It's also very hard to replicate this radiation. So someone, in the audience may ask, well, can you do these experiments on the ground? Do you really have to fly them into deep space? Can you go to a particle accelerator, generate these ionizing radiation particles and do your studies there? And so the answer is yes and no. Yes, because we are actually doing that. We're doing a lot of radiation studies on the ground to get ready for the data that we're going to get in space. So before you get to space and collect that data, you have to make sure you understand the system. And so you can use doses that you specify, and you can get the data, and you can look at the biological response. You set up a database, then you fly your experiment in space, then you collect your data from space. You then compare it with the data that you have on the ground, and then you say, oh, you know what? The changes that we saw on a 1-year duration mission around the sun gave us biological damage that was roughly equivalent to when we bombarded ourselves with so many grays of iron on the ground. So those are the sorts of ways in which you can make a biosensor to calibrate the biological effect of radiation. And also, as I mentioned, lack of gravity, which only you can reproduce that only in space, because it's very hard to do that on the ground. It's hard for us for more than a few seconds to recreate the lack of gravity. And that can confound. And so when you really want to know how a biological system will react in space, you have to do some experiments in space. What are some of the challenges? So I talked about doing these experiments in space, but really, is it like doing experiments like you do in your labs? You go to your bio lab or your chemistry lab, and you have your protocol, you have your lab bench, you have your reagents. How does it work in space? Well, there are lots more challenges, as you can imagine. So for one thing, you have this long pre-launch period. So I, for example, will send my experiment to Florida, to Kennedy Space Center, and it will sit on the pad for 6 months at ambient, meaning at whatever temperature and humidity is in the environment around it. And then it will launch into space. Then we will be able to activate, then we will get power to activate our experiment. Then we can add the media, the nutrient media to our cells to grow them. And so in this whole period, your cells have to stay alive and your media has to be fresh and workable. And so all of that is very different, from when you do experiments in the lab. And so all of that has to be tested before you get up there to make sure your experiment's going to work. Also, in these satellite missions in deep space, you don't get any samples back. And so you have to be really clever about doing your experiment so that you can get all the data back by radio or telemetry down. And then you have to be able to make your interpretation in a robust fashion based on the data that's coming back down. And so all that comes into the planning of an experiment as well. So you can imagine it takes years to get ready for each of these experiments that you do. And then, as we mentioned, of course, you can't do any testing with humans with radiation. So your best bet is to use these simple model organisms, which the National Institute of Health has said, for example, are genetic systems like yeast, Saccharomyces cerevisiae, the same yeast that you actually use to make bread, is a very useful tool to scientists because its genetics are very well characterized. And so I'll tell you about the yeast experiment we're doing, and I'll tell you a little bit about fruit flies as well. So yeast, right? Now, this is, of course, a hugely blown up picture. The yeasts are microscopic. They're just a few microns in diameter, so you really need a very high-powered microscope to look at these cells. And then there's the human. And there's a 1 billion year separation in evolution between the human and the yeast cell. But you know what's really remarkable is that in spite of that, the structure of the cell is very similar. So they are what is called a eukaryotic cell. And some of you, I'm sure, in biology either have studied it or will study it very soon, where you can learn a lot about the function of a cell from a yeast to a human. In fact, there was a study published a couple of years ago in the journal Science, where, and now that you guys know this concept of the DNA sequence and the genes that I talked about, what they did is something interesting. They took the yeast cell, they deleted one by one, they deleted very important genes in the yeast, which normally when you get rid of them, the cell will die. What they did is they took that cell and they took genes from the human, one by one, to see if they could complement the loss of the yeast gene. And 50% of the time, they were able to complement the loss of function in the yeast cell with a human gene. So in other words, what that shows you is that there is a lot of similarity, and human genes can often function in a way similar, even in a yeast cell, a cell as simple as the yeast. What's even more remarkable is that if this you could do in a random fashion. If you, on the other hand, took a more deliberate approach, and let's say you said that I'm going to take the genes that are responsible for digesting the sugar or the glucose that I ate this morning, and that's being enzymes that are digesting it in your body. You take those genes and you take the same analogous gene from the human and you put it into the yeast cell. Then 80 to 90% of the time, the human gene will be able to positively complement or substitute for your yeast function. So I'm giving you this as an example to show you that even a simple system, as simple as the yeast, has a lot of functional similarity to the human. And this is something often for those of us who don't think about it on a regular basis, it blows your mind, right? To think that something as simple as that has so much similarity functionally in how your cells in your body function. All right, so what is this biosentinel mission that I've been talking about? What exactly does it look like? What is it all about? So we're going to use these cells that I told you about, and we're going to, and they have a very well characterized DNA system. We know all about their genes, and we are going to use it as a tool to quantify, to measure the amount of DNA damage that this radiation environment is going to impose on the yeast cell. I mentioned to you again, why are we doing it in space? Well, I told you we couldn't recreate that environment exactly on the bench on the ground. We're doing a lot of studies on the ground, but eventually you have to do the study in space. So we're using lab-engineered yeast cell, which will sense the damage that occurs in their DNA. We will be able to quantify it, and the yeast will sit in a dormant, dry state for many months till I have the time and the opportunity and the power to start my experiment. I told you already why budding yeast. It's a very useful system. It has similarities, and more importantly, it can be stored dry for a long period of time till you're ready to activate your experiment in space. You can't do that with human cells. You can't do that with a lot of different cells which have much more complicated ways in which you have to look after them. The yeast is a nice, robust system which will withstand all of these logistical challenges of doing a space flight mission and yet give you valuable information at the end of the day. Why satellites? Well, I'll tell you about satellites now, but later I'll also talk about the International Space Station and other platforms. In this case, we're using a satellite because we can go into deep space now. You also, as a result of using satellites, they're small. So it's somewhat more manageable in terms of how much power they need, how much capability they need. You can get more frequent access into space because it's easier to launch a satellite than to launch an entire rocket, which is expensive and time-consuming to build and doesn't happen as frequently. It's a very good educational tool. So a lot of students actually work through various NASA internship programs in the lab to help us come up with some of these experiments. And there's a lot of value because you can test new and novel and creative technologies to see if they work with minimal impact to anything. And then if it works, you can then use it on things like these heavy lift rocket capabilities and so on. So this experiment will actually be the first biological study that we will fly beyond low Earth orbit in over 40 years. So the Apollo missions back in the day that went around the moon, landed on the moon, those were the last time that we've been in deeper space with this high radiation environment. And this will also be the first CubeSat So our experiment will literally be this big only, and it will be a fully automated self-sustaining system. And so the idea is, this is the Space Launch System Exploration Mission 1. It's the heavy launch capability that NASA is building now to take people and payloads into deep space. And we will be one of 13 boxes here in this adapter area. And what will happen is, let me just go back to this one. What happens is when the EM1 goes around, so it's going to be the maiden voyage. EM1 stands for Exploration Mission 1. It'll be the maiden voyage of this heavy lift capability that will go around the moon and come back. It's to test that everything works and is safe. And then when you see that that's the case, then humans will take this rocket and go into space. But we will actually piggyback on the rocket even before humans get on it. And so on this very first maiden voyage, we will be there, we'll piggyback as a little fly on the wall, and we will get released when the EM1, the Orion vehicle, is coming back to Earth. We are going to get, like a slingshot, we're going to be thrown out, and we will then go around the moon, and then we'll go towards the sun, and we will be collecting data and sending it back down for almost 1 year. So after we launch it, 6 months to a year, we will be getting data back from our yeast cells in space. This is what our hardware will look like. This is the box that I told you that's this big. These are the 4 solar panels that will point towards the sun and get us, so it's very eco-friendly. It will get energy from the sun and will power up everything that we need, all the software, all the media moving around, all of that will be powered by the sun. Now, what does this look like? Again, remember I told you that in your bench, you have a little plate with wells, you put these cells in there, you put media, you grow them, you put it under a machine that reads them out. Simple. Well, in space, unfortunately, not that simple, right? That same experiment that you do, and there'll be 18 cards with 16 wells each, and each of these wells will have 100,000 yeast cells in them. And we put those yeast cells in them, and then maybe every month or so, we will activate 2 sets of cards. So we'll activate, 32 wells with 100,000 yeast cells in each of them, and we'll watch them grow. Now, this is what the thing looks like. So if you took this thing that I showed you where the yeast cells are, and you cut them vertically like this, then what you would see is, of course, this is blown up hugely. This whole thing is just this big. And what you see is your yeast cells are in this well here. You have LEDs, so LEDs are light-emitting diodes, so you can have specific wavelengths of light that you put through this well with the yeast cells in them. And you have a detector board here, which will measure the changes in the reading of that light as it goes through your cell. And I'll tell you in a second how we do that measurement. But the idea is, again, I want to show you is that, again, simple. On the ground, you just put yeast cells there, you put food in there, and you grow them. Well, here in the space, this many layers of plastic and other materials are needed because you have to heat them. You have to heat the cells right when they want to grow. And you have to have an optically clear path so that LED lights that you're relying on to do your readings can go through. You have your LED emitters, your light emitters, you have your photodiode detector arrays at the bottom that will capture the light. And all of that just gives you this one simple little box which will have 16 wells itself. So how does it work? And again, this is just a little detail for your interest. How exactly are we doing the measurements? What are we measuring and what data are we sending back to Earth? So we utilize something called Alamar Blue, which is a dye. That's an interesting dye. It works a little bit like many of you have probably used litmus paper in the lab, for example. It changes color when pH changes. So this is something similar to that. And what happens is when cells are growing, in fact, even when you are sitting here in the chair, you're not running around, you're not moving. Even when you're doing that, every cell in your body is active. The cells in your brain are probably even more active because you're thinking, you're seeing, you're listening, and so on. So what happens is every cell is constantly generating energy and is secreting stuff into the media. So this dye detects that stuff and changes color. So it goes from blue to pink, pink to clear, as the cell keeps growing and and doing its thing within the hardware. So what we have, those three LEDs that I told you about, there are three different wavelengths of LEDs. Now this red wavelength, for example, measures the amount of the blue color. So as you lose the blue color, you can see this one gains pink color. So the green LED measures the increase in the pink color. So as your cells grow, you lose the blue color, you increase your pink color. Those are the readings you're getting from your spacecraft. And then the third measurement is for optical density. It simply measures the turbidity or the density of your cells. So it knows whether your cells have doubled or tripled and so on. So it measures how many cells you have in your system. And so just with these simple three measurements with light, you get some very valuable information. You get what is called a growth curve. So in other words, you get to measure how these yeast cells are going to grow. And so this is an experiment, for example, with increasing doses of radiation, which we know, right, as we talked about earlier, are harmful for cells. So as you get more and more radiation, this purple has more radiation than the blue. In fact, the blue has no radiation. And then as you increase radiation, your curve shifts to the right, which means that you're getting slower and slower growth of those yeast cells. And we quantified this, and this is the measurement then that we use. So just from the LEDs, you get to get very important information about how your cells are going to respond to radiation. Now, the other thing that we have in this paper, so I told you about the biology and the yeast cells and how these LEDs are going to help us get information from them. We also have a second component to our experiment, and that is we have a physical, what is called a dosimeter. It's simply a physical device that measures, so it has pixels, it's a solid state device. It has pixels. Each pixel will monitor how many particles of radiation are coming, when are they coming, and what's the energy of the particle. And with that information, we can map a spectrum of the radiation that's hitting our experiment during our mission. And so what is really interesting here is we'll have data from a physical dosimeter that will tell us how much radiation hit our spacecraft, and we'll have the biology with the yeast cells that are growing, where we look at shift of growth, and we will correlate the two so that we know that okay, in month 6 of our mission, when we were so many thousand kilometers away from Earth's surface, we got this much of radiation, and this is what the biological response was. And that's how we're going to use the system to calibrate, so use it as a biosensor to calibrate the biological effects of radiation at different altitudes and different places in space. As you can imagine, this is a pretty complicated, it's really literally this big, but it holds a very complicated set of things because this little box has to be completely self-contained. It has to do science, it has to be powered, it has to activate your yeast cells when you want to, it has to store the data from all of the science that you're doing, it has to be able to talk to the antenna on the ground and radio all of that information down periodically. It is commandable, so even though there's a software program that runs all of this automatically, if we see something happening that we don't like or that we want to change, we can command the spacecraft from the ground, from certain places on the ground, to say, no, change that, don't start these 2 sets of wells yet, wait another month and start the experiment then. So all of these capabilities are built into something that small. And like I mentioned, we could go to the moon. We are already going on the M1 around the sun. We also have planned a mission in the next year or 2 to go around, to go to the ISS. And we can use this then to go to other destinations, other planetary systems. So to conclude this first bit of the talk, I wanted to just mention that you can do biological studies in low or near Earth orbit to show you, and well, the studies that we have so far show us that it is important to study the effects of this radiation environment. I've also talked to you about how you can do real science using these satellite platforms. We can use simple cells like yeast cells and get very effective information on the biology from them. We can compare the effects of radiation in different places in the solar system and beyond. And the heritage of what we learn from here will really help enable these larger, heavier lift missions in the future. Now I'm going to shift gears just a few minutes for the last part of the talk. Oh, actually, before that, to thank all of the people that I work with. As you can imagine, the engineers, software engineers, project managers, scientists, whole group of people. And it's really fun. It's a very interdisciplinary team that we work with, and it's great fun. So I'm going to talk to you a little bit now about fruit flies. So these little things, which, you know, over here it's hugely blown up, but you can imagine there's this tiny little speck that you see buzzing and annoying you and flying around your body. That fruit fly has so much similarity between humans that often that surprises people as well. So when you look genetically, there's almost a 75% match between the DNA and the genes in the fruit fly and those that we know are important in the humans. So there again, you know, that's a very humbling thought again, you know, where we think that humans are so much better than some of the insects. But really, when you get down to the brass tacks, we're not that different. So for spaceflight particularly, the other benefit of using these small model organisms is that in a box this big, I got back, a couple of years ago, for example, I got back 4,000 fruit flies. Now, what is the benefit of that? So let's think about that for a second. We know that astronauts have gone to the moon and back, right, from the Apollo missions, and they've survived and everything. So we know that chances are nothing highly catastrophic is going to happen. However, we also know from some of those studies that I showed you that there are going to be changes in your biology. So they could be subtle, but very important. So when you have a subtle change, think about it. If you send 4 astronauts or 6 astronauts, you know, let's say someone is from India, someone is from the US, someone is from Russia, someone is from Europe, someone's a male, someone's a female, someone's 40 years old, someone's 20 years old, someone's 30 years old. It's very, very difficult to compare data between those subjects and come up with an estimation of what will happen in deep space. But when you fly fruit flies, all of which are genetically identical, and in a box like this big, you can bring 4,000 flies back, you can imagine that your statistical power on doing the science is very strong. So many of you, you know, in your labs, you will be doing standard deviations and standard error of the mean and so on. And you know that in your data, unless you can get data that's tight and reproducible, you can't believe it, right? And so that's where having numbers is so powerful for doing science experiments. So one example of a mission that we did apply a few years ago, we showed that the flies have a change in their immune system. Now, what is the immune system? We all have it and we all rely on it. Our immune system is our blood, is one of the components of our immune system. It's what helps us fight infection. So when you have bacteria coming into your body, the immune system, the blood, gets into the act, fights off, wards off these bacteria, and that's when you feel better, right? So the immune system is critical. And the flies actually have an immune system that's very similar to the human immune system. So one of the things we showed from our recent mission was that when you compare ground to space, and sorry, it's not very clear to read, these are the space-like samples and in blue are the ground, there was a marked reduction in the number of blood cells. And these blood cells are the ones that will help you fight an immune system. And again, you don't need to really look at these graphs. I'll just describe to you what we saw. And this is, if any of you are interested, this paper is in the open literature. So check it out if you're interested. But these green things are your blood cells, and the red things are the bacteria that your blood cells can capture and kill. And that's how you feel better. So the flies have the same thing. And what we found is that flies that came back from space were much worse at being able to, their blood cells were much less efficient at capturing and killing the bacterial cells. And so these are some of the things that, for example, are important for us to study. And then we did the molecular biology. Remember I told you about gene expression and DNA? So we did follow-up studies and we found that many of the genes that are important for the immune system were depressed. And that would explain why the blood cells were not working that well in fighting the infection, and so on. So in other words, here's an example where again you can use the fruit fly for a system that's important to us, like the immunity of an astronaut, to study what would happen in the space environment. And once you understand the molecular biology of which genes get into the act and which genes are depressed, you can then think about countermeasures and how to rectify the situation and make it so that the astronauts don't have any negative effects in spaceflight. And so the last example I'm going to give you is how we have actually just got samples back from a mission that we flew a few months ago, where we used, this is the body of the fruit fly, where we used the fruit fly actually to study heart function. And I kid you not, a lot of people look at me and say, well, you've got to be joking. Really, the fruit fly has a heart? And the fact that the heart actually functions similar to the human heart, it really blows the mind. But here in red is the tubular organ, that's the heart, the cardiovascular system. And this over here is a muscular pump which actually flows blood to the body. And so when you look here, you find, like if you did a study on the human population, so let's say you looked at the older people in any population, what you would find is that there's an increased rate of arrhythmia in the heart. So your heart normally beats at a very steady pace, but as we grow older, we are more likely to have irregular heartbeats. Now watch what happens. This is a fruit fly heart now that I'm going to show you. So this is the tubular organ that I was telling you, and look at how it's beating, right? So it's beating like this, and the blood is flowing through this tube and flowing to all of the organ systems in the fly's body. Now this is a young fly. This is a 1-week-old fly. So it's the equivalent of a 10-year-old child. Now watch what happens when I show you the video for the 7-week-old fly, which is the equivalent of a 70-year-old adult. And do you see that? Sorry to be in the way, but do you see you see how irregular that is compared to the, see that? So this, it almost exactly recreates what you see in the human population. So in other words, aging and how it affects heart function is very similarly conserved between flies and humans. Drugs and hormones that work in the humans work in flies too. Clofilium is a drug that's used sometimes to slow down the heartbeat, and those of you in the audience who are doctors are familiar with this. And if you look on the time axis and you look at the beating of the heart, you'll see that the heartbeat slows down with clofilium. And this is in the fly heart, the same drug used in humans. And similarly, hormones that affect heartbeat in humans affect that in the fly as well. So this experiment that we just flew, that we just got back the samples from, we flew wild type, or what is called normal, or wild type of control flies. And I've told you that flies have similar genes of proteins to human hearts. They show cardiac aging effects similar to humans. All flies from a genetic line are identical. You can compare populations of wild type and mutant flies to understand what's happening. And what's interesting, again, in this experiment with flies is you can set up mutants. So we've also set up a mutant which is predisposed to arrhythmia. So even without sending it into space flight, they are more likely to show these arrhythmias and these irregular heartbeats. And so what we've done is we've sent it into space so that we can bring it back and measure and see, does it make the condition even worse? And therefore, is this gene involved in the function in space? And this is one I wanted to tell you about a little bit, is because the human equivalent of this gene mutation is called the long QT syndrome. So some of you may be familiar with the fact that, you'll read in the papers that there's an athlete who's, 20-some years old, a young athlete who's in the peak of their health, and yet one fine day they drop dead and of a heart attack. And that sometimes, or very often, is from an undiagnosed condition called long QT syndrome. And the fly has the exact same mutation in it. So you can study a human condition in the fly, for example, with this mutation. Another mutation that we flew is for a muscle mutant. So in this particular case, as you know, in space flight, there's no gravity, right? And we take gravity on the ground for granted. But when you don't have gravity, your muscles will start to waste, and your bone density will reduce as well. And so we have a mutant that has a defect in muscle, any muscle function anyway. So it's what is called, it will be a little more sensitive in space. And we're going to study what happens to those. Does it make the condition even worse when they come back down? So in conclusion, it's important to assess the biological effects of the spaceflight environment, including the lack of gravity and the fact that there's a higher background of radiation on a biological system, and that simple genetic model organisms, and I've given you two examples, the fruit flying yeast. Other people work in mice, other people work with worms, C. elegans, and so on, plants, and so on. And the data that we collect from these are absolutely critical in helping us prepare for long-term exploration in space. These are some of the people in the lab that have been key in doing some of this work. But most importantly, thank you to my family. Without the love, support, and being there, for me day in and day out, couldn't have done it without them. My mother, Dr.Sharmila Bhattacharya, who's sitting in the front row of the audience, I owe a huge, I can't even really ever quantify that, what I owe to my parents. My father, Shukdev, my brother, Shivaji, my husband, Raja Buhataputta, my sister-in-law, Lopa, and my daughter, my 20-year-old daughter, Tara. So with that, and of course to my extended family and close friends and all of you who are also my friends, and to the Birla Museum and to the American Center. So thank you all, and with that, I'll take questions.
Okay.
Hello. Any questions? Dr. Bhattacharya is ready for questions. So students, if you have any questions, please raise your hand. Yes.
Are there microorganisms living in Mars? Oh, good question. Very good question. So there are space scientists who are looking at that. The answer at this point is that there may well be, yes, because it is thought that there was water at some point. Some of the environmental conditions could have allowed that. So in fact, when this EM1 rocket allows us to go into deep space to collect samples, those are exactly the kinds of questions that we'll be looking at to see. And also remember, there probably is or was some sort of life form, but the challenge will be recognizing what it is, because it may look very different from the life form that we have on Earth. Because remember, and so, yeah, that's part of the excitement of doing the science is you find out what it is. But yeah, great question.
Thank you.
Thank you.
Please introduce yourself first and then ask question.
Yes, tell me what school you guys are from, or
He was from Cambridge School. Your name? Shankho from Cambridge School.
Thank you, Shankho.
Thank you, ma'am. We are very much privileged. We are coming from the Sri Sri Academy. We are very much privileged by observing your presentation. One of my question is that something related to queries.
Yes.
Can cells, since you have already talked about, spoken about that genes from the Drosophila and your Saccharomyces.
Yes.
So can stem cell therapy can be done with these even in the ground or in the space?
Excellent question. Yes, I think there's no reason why not. Now, the thing with stem cell therapy that we have to keep in mind is at this point still, it's pretty complicated. And so it needs a lot of tools. You need to monitor its progression. But eventually, I'm thinking, maybe not even a few decades from now, but some years from now, hopefully it will become more routine and easier to implement. And certainly that's exactly the kind of thing that we would be interested in. Because if you are mutating something with radiation, stem cell therapy would be the ideal way to counter it. So thank you for bringing that up. Yeah, that's absolutely the way of the future. And our challenge will be, like I showed you, to do an experiment on Earth. You just pipette them in. Here we'll have to just work out, but everything's possible. So I know with the way technology.
Right now, scientists are doing stem cell therapy on the space.
Right now, no, but colleagues and friends of mine are doing experiments with stem cells on the International Space Station, where they're looking to see how do stem cells respond, how do they respond to radiation. And so, yeah, so that work has definitely begun. But at this point, as far as I know, no substitutions, etc., have actively been done in space. But the groundwork is being laid to do that stuff. But yeah, so great question. Yeah, absolutely. Thank you for coming.
Will it be harder to live on Mars than on Earth?
Yes, yes. Something you want?
When I got from the Cambridge school.
From which school?
The Cambridge school.
So yes, I'm afraid that I would have to say at this point that it would be much harder to live on Mars than on Earth. And that's good. But just think about, our ancestors way back when you're thinking of cavemen and so on and so forth. It was much tougher for them to live than it is for us today, right? So humans find a way to kind of, make themselves comfortable where they are. So with time, things will improve. But at first, it certainly will be a challenge because you'll have radiation, the environment is different, your daylight cycles are different. So yes, but yeah, good. Your generation is who's going to get us there. So keep thinking about it. I think there was a question up front here for a student. There's a question here.
Good morning, ma'am. I'm from Sri Academy.
Okay.
My question was, can we build a shield or a barrier with which humans can prevent gene mutation in space?
Excellent question. Yes. So, and I'm glad you brought that up because I touched on the issue of shielding being difficult, but I didn't really finish that story. So thank you for bringing that back. So the answer to that would be, for this galactic cosmic radiation, the only material that really seems to help, and not completely, but to some extent, is hydrogenous, heavily hydrogenated material. So water is a good shield, surprisingly. But the thing is, how much water are you going to carry? Because your heavy lift capability has to take fuel for propulsion for the rockets, it's going to take all this other equipment. So you don't really have the mass and volume capability to take large amounts of water up to shield yourself. So that's one thing. However, it's a very good question to think about because there's some other materials that you can use on the planet that you go to. Let's say you go to Mars. There is in fact, if there's water there that's usable or that one can tap into, you can see that humans might be able to build a shelter surrounded by water over there, which will help shield them. So that's one thing. Let's say you go to the surface of the moon. The moon has something called regolith, which is basically lunar dust. What you can do is, it's thought, and again, all this has to be tested, but it's thought that this lunar dust can be used to build something almost like cement or concrete that you can use around you as a shelter to protect you from the radiation. So there are these sorts of things, and people are thinking about it because as we plan to go into deep space, you have to think about these things. How are you going to protect your, when you're sleeping at night or whatever? How are you going to be in a more shielded environment? And so there's a lot of studies being done by material scientists and stuff who are looking at things like that. So excellent question. Yes.
Yes.
This each cell which you are using for it, there will be each many of different types also. So what particular each cell you are using?
Yes, thank you. Thank you for asking that. Yes.
Another one more. Yes.
Yeah. So we're using Saccharomyces cerevisiae, which is the brewer's yeast that's well characterized as a genetic model. So it's Saccharomyces cerevisiae, budding yeast. Yes.
At one time you mentioned about the effect of smoking. So what is that particular thing? You mentioned in your slide for smoking cigarettes, the effect.
No, I don't remember mentioning.
So you mentioned there, because written there also, you mentioned also, but forgotten.
This is what old age does. You start forgetting what you just spoke about.
What is it?
You know, so smoking and that are you talking about cigarette smoking? Is that what you're referring to, cigarette smoking? Or?
I don't know, you mentioned there.
Okay, yeah. So maybe, you know, there was some misunderstanding on that front. But in general, though, the toxic chemicals like what is present in cigarette smoke can be a carcinogen. So, on a slightly different note to the kind of radiation I talked about, in addition to radiation that can damage DNA and cause cancer, you can also have environmental toxins. So cigarette smoking is one of those environmental toxins that can also cause cancer by damaging cells because they affect the DNA. So maybe in that context is where, you know, as an environmental toxin would be how smoke would affect.
Now the solar radiation at different levels, it is different. So how that also affects in this matter?
Yes. So I think the biggest thing is going beyond the Earth's magnetic field. That's the biggest difference. Once you go beyond that, whether you're near the sun or whether you're near Mars, the radiation environment is going to be pretty similar. It's going to be very damaging, but very similar. But from here to the Earth's magnetic shell and beyond, the distance becomes very important. And that's why some of the damage we see on Earth is very minimal. But then you go to the International Space Station, you see more damage. And then as you go higher, you go to the moon, you see even more damage. But beyond the moon, you'll have that same radiation environment more or less everywhere. So it evens out finally. Yes, you're welcome. Yeah.
Ma'am, I'm from the Future Foundation School. So you just talked of GCRs and SPs, right? And you talked of the shielding thing just now, but I think about the detection thing, I have studied somewhere that GCRs can be detected by means of gamma rays. And SPs, you talked that they are not possible to detect because they can hit all of a sudden. So why is it not possible to detect SPs?
So thanks for clarifying that. So it's not that it's hard to detect. You can detect GCRs and you can detect protons with a dosimeter. In fact, that's what we're doing, right? Because we have our LET spectrometer that's going to measure the amount of this radiation. So you can certainly detect. What's difficult in the case of GCR is shielding. As she brought up, it's so heavily penetrating, it penetrates material. So that's the GCR caveat. On the proton side, what's difficult with protons is prediction. Not detection, but prediction. So in other words, you are going about your merry way, doing your extracurricular activity, fixing this, fixing that, when you're doing a spacewalk, but all of a sudden an SP hits with a radiation dose that's 1,000 times what it was an hour ago. That is what you don't know. So the predictability, it's what is called a stochastic event, where predictability is very, very low. And so that's the challenge, not detection. Detection of both particles is easy. But what do you do to prevent getting hit by that is what is difficult in both cases.
From this one follow-up question, like GCRs, what can be called the main cause of the formation of the root cause? Like what does it form from the GCRs? Like
yes, so what it is particles like iron, for example, it's heavily charged iron. What happens is they physically come in and they mask the cell. They form what is called these reactive oxygen species, so hydroxyl radicals.
this is the nascent oxygen that you're talking about.
Exactly. And those are what further damages the DNA and causes the double-stranded DNA breaks. And that's what's so damaging to the cell. Yeah, absolutely.
Okay, ma'am, I have
Yeah.
okay.
No, it's okay. Go ahead and mention that. Yeah.
Okay, ma'am, I have a question. I'm in Peter Foundation School and my name is Pankaj Chakraborty. So I was going to ask you that how can we use the particle accelerators in the ground?
Yes, excellent. Yeah. So in fact, we do use particle accelerators. For example, at Brookhaven National Labs, there are particle accelerators here. In fact, Bhabha Atomic Research Center in Mumbai. There are some in Japan. There are some in Europe, and so on. You can use it and you can generate these particles. So in fact, a lot of our space, our ground-based data is at the particle accelerator.
We can generate these particles on the Earth, but there is effect of gravity on the Earth. So
exactly. So that's why you need to do the experiments in space, because we can't get rid of the gravity on there. That's one. The other reason is even for the radiation you need to do it in space is because in the particle accelerators you can generate one particle here, one particle there. But in space you're getting constantly bombarded with one iron, one oxygen, one silicon, one, and you can't simulate that on Earth. When I go to Brookhaven, they give me 1 hour's time with the iron radiation beam, and my postdoctoral fellow goes and does the experiment then. But it's impossible to recreate that richness of the radiation environment that you would be bombarded by in space on Earth.
is there any effect of the what is called the gravitational waves on the human body?
That is a little bit so just gravity as a force is what you're asking?
No, I'm telling you the waves which are gravitational.
Yeah, so that's a more physicist question that you'll have to find a physicist to talk about. For us, I can tell you as a biologist that the way gravity definitely affects you is that it causes, with no gravity, you would start losing muscle mass, you would start losing bone mass, you would start seeing some of the changes I talked about with the immune system and so on. And so gravity is a force, certainly, that's very, very important. In terms of gravitational waves and the subtle detection at those levels, whether biology is affected by them or not is a question no one's looked at.
You're
welcome. Thank you. Yes, Audrey, can you please have a question here in front? Yes. Of course, all questions about science. This is getting from the same question. People asking about safeguarding. Yes. When you send the astronauts into space, have you found out the effects of radiation on each astronaut? Have they been affected at all? What are the safeguards that they've used? That's one. And the second thing you told us was about the lunar dust that you collect, and about the water from Mars, perhaps. Now, have you collected those specimens? Has it brought them back? And have you tested the effect? Yes. So good question, both of them. So let me tackle the first one first. So so astronauts, there's actually, Johnson Space Center, the NASA Johnson Space Center in Houston, in Texas, they are the ones who collect astronaut data all the time. In fact, from the time they're recruited as astronauts to when they fly to after they come back, there's constant monitoring of their health, their blood work, their heart function, everything is tested to death pretty much all through. So there is a lot of data on astronauts that have flown for different durations. And there are some papers in the literature that do report a slight increase in the incidence of cancer. But again, the challenge is that different people that are flying are different ages, they're from different countries, they eat different food. So sometimes it's hard to make generalizable conclusions from the human data. But at least so far, it does look like there is an effect on astronauts in terms of, for example, cancer incidence. I'm sorry? They don't run the risk of getting cancer. They do. A slightly elevated risk of cancer, they do, yes. And again, it depends on how long they've been in space, how old they were when they went into space. So all of those things factor in. And then your second question about has anyone collected, lunar regolith and so on and so forth. Yes. So the answer is there are some moon dust samples actually that's heavily and carefully protected because it's very limited amounts. So from the previous mission, and then recently we haven't been in a long time, so there have been no new collection. But there are plans to go back to the moon, and then we hope to get more samples. Because even to do science, right, you need enough samples to be able to do the science. So that's where again we're looking forward to doing more missions. What about water in Mars? And water in Mars, yes, definitely no one's collected those samples. Yeah, but again, you can have robotic missions maybe that go into Mars and start collecting. But again, think about it. On Earth, you take a little scoop, you take some soil or water, you put it into your container, and you bring it back. It's a whole different story there because how deep you have to dig to get water. What tools do you use? On Earth, we use gravity a lot for a lot of things, and we take it for granted, like when we're drilling something. But when you have, less gravity, how are we going to do it? So it hasn't been done yet, but I'm hoping in the future that it will.
What's the ultimate aim of scientists?
Yes.
Where do you want to go ultimately?
The ultimate goal, as we talked about, is to go into deep space, to go into all these different planetary, and to be there safely.
With human beings,
Yes, and be comfortable there, go there, yes. And now space tourism is this commercial. So having the commercial sector work with NASA, so SpaceX, Elon Musk's company, Elon Musk who does the Tesla cars and so on, having those and Orbital and Virgin Galactic and all these companies has really helped actually, because it increases the because any space agency has a limited amount of funds, and so there are a limited number of projects you can do. But when the commercial sector gets into it, there's more frequent access to space. Exactly, Virgin Galactic is Richard Branson and all of those people. Yeah, so that's where Blue Origin and all of those companies. That's what I think will make a difference, and that coupled with space agencies, we will have more frequent access into space. Oh, that's a tough one. Every time. And I'm not going to say how many years that will be, but today, if I had to say, maybe I would say it would take a few decades to get there. But often, in fact, I was in a workshop recently where they were talking about how quickly technology is developing. And this is the generation who are going to make that difference in technology development. And so every time we make that leap in technology, we leapfrog decades ahead. So it's hard to predict at this point. It may be sooner than we think. And I hope it is. Yes, you're welcome.
Is there any chance for life in Pluto?
Is there life on Pluto?
Yeah.
Because Pluto is so far, we actually haven't had that many studies, as far as I'm aware, that will tell us. More of the studies have been with Mars or with the Moon, and so on. Maybe the life will be a different form, very different from the life we have on Earth. It's possible. Maybe we won't recognize it when we see it, right? Because we're used to this limited life forms that we see over here. So who knows? There may well be. Sure. Yes, even extremophiles, exactly. Yes. Yes. And whenever, I do. It with respect. Exactly. Yeah.
I am Orlando from Cambridge School. My question is there liquid water in any planet except Earth?
So right now, there is thought that there may well be, on Mars. In fact, some of these robotic, these voyagers and stuff are looking at spectra. They're looking at surfaces and they see what they think is ice. So the answer is quite possibly yes. And where else it might be, we will wait to find out.
Also, what is the difference between a dwarf planet and a planet?
Say that again. What's the difference between a what and a what?
A dwarf planet and a planet.
I'm not sure I'm actually even hearing the question. Different planets, is that what you're asking?
I'm asking what is the difference between Pluto is a dwarf planet,
yes,
and Earth is a planet.
Right.
What are the differences between them?
So again, you're asking an astronomy question, but those, my understanding is based more on the size, right? And because bodies, celestial bodies the size of Pluto are now thought to be much more common than they were back in the day. Back in the day, Pluto was considered one of the planets simply because it had been visible. But now, and this is where we were talking about advanced technology, now we have better telescopes, and again, this is completely outside my area of expertise, but with increased power of telescopes, you can see more bodies out there, more planetary bodies. And so if you were to call Pluto a planet, then you would just have so many more that you would have to name. That somewhere the astronomers, a different group of scientists, have to find a cutoff as to where they draw the line for what's a planet and what's not. Thank you.
Good afternoon, ma'am.
Yes, good afternoon.
My name is Vishal from Kalyan Community College. So, as we know that mutations play a great role in evolution. Okay. So, we know that the reproduction rate in case of saccharomyces is much higher than human beings. So, the frequency of mutations will be also higher. So, is it possible to get those mutations that replicate some positive effect in case of their survival into human beings?
Absolutely. In fact, those are exactly the kinds of studies that we're interested in. Because once you know what the changes are, and then you know what the genes are that are changing, then you can start thinking about, and this is where I think this gentleman brought up the stem cell question. You start to think about how can you positively, or how can you send up baby mutants who are better. For example, they may have increased expression of an antioxidant gene, which will reduce that oxidative stress and the reactive oxygen and the superoxide radicals that we were talking about. It would reduce that and then make the damage from the radiation better. So absolutely, yeah, that's exactly why we're doing some.
So is there any kind of possibility of antidotes for for preventing the radiation damage?
Yeah, I think the key for one of the keys may lie in antioxidants, either in antioxidant supplements or genetically engineering antioxidants in a certain way.
Another question I have. So we know that deep space exploration is much more, it takes lifetimes for human beings.
Yes.
It's the scale of human in case of the exploration. So is there possible ways to hibernate certain parts of the cells of the nucleus so that we can extend the lifetime of human beings?
Yeah, so in fact, some people, some of my colleagues are actually looking into some of that. Is just like bears have hibernation, is there a way for us to find some biological process whereby we can make I mean, at this point, it's still science fiction, but it may well be possible because we know animals on Earth like bears do hibernate, and they are very well protected during that time. So if there's a way to do it for humans, it would be fantastic. Yeah.
Thank you.
Thanks. Yeah, thank you very much. So you should let me know whenever you need to. One last question.
On plants also, plant cells also, is there any study made? According to the old Indian science, there are 27 plants of 27 planets. So how they belong to those plants?
Yeah.
So is there any study made about that?
Not that I'm aware of it yet, but maybe Indian Space Research Organization will do that.
Okay, that's
so one
last question.
Yes, please. Good afternoon, ma'am. Good afternoon, yes. Ma'am, it's obvious that the Earth has protective layers that blocks out harmful radiation. Has anybody discovered that other planets have such layers that block out such radiation? Very good question. And having that magnetic field, so a lot of that magnetic field is thanks to our gravitational field. So chances are yes, because many of the other planets, for example, do have some amount of gravity. But for example, Mars has gravity, but it's a lot less than Earth. Moon has even less. So many of these may have much less protection than the Earth's surface. But that's an excellent question. And in fact, the other question is, can you utilize this principle of having this magnetic protection? Can you utilize it in an artificial way? Meaning, can you create this around your habitat to help prevent it? Can you have can you create a magnetic field around your habitat when you're in deep space to counter some of this radiation? All of that is being looked at this point, because it also has to be safe for the biology, but at the same time, it's got to be feasible in the environment you're in. But that's an excellent question.
Yeah, thank you, madam. We have to end the question and answer session right now, because if we continue and go on forever, I know we have a lot of questions, But Madam is available in YouTube and as well as Facebook, so you can always communicate with her. And as a token of appreciation, we will ask these two students to felicitate the speaker. I'll ask Director, Mr. Islam, kindly go on stage and hand over to the speaker and token of appreciation for Birla Museum.
It's a pleasure and it's so exciting. On behalf of all of you,
thank you, Madam Bhattacharya, for your excellent speech. A round of applause for the speaker. For coming so far from NASA and enlightening about us on many things we didn't know. Thank you, Madam. I'd like to thank you, the schools, for attending this lecture, the students and the teachers who have taken the pain to come here. I'd like to thank the American Center for organizing and giving us the opportunity to host this lecture. Thank you once again to all here. To the students who have received the full coupons, please redeem it from the entrance from where you are registered. Kindly take the coupon and get your tuition coupon from there. The students who haven't not as yet received